as before, please could those of you who are really at the back move a little bit forward so it's easier to see you and interact with you. The second announcement is that for those of you who have to collect some travel reimbursements, the person will be out here at 3 o'clock after the session, after the afternoon participant talks. By 3 p.m., you will have someone with a desk over there who will be able to handle your reimbursements before the weekend. Okay? Did I have anything more? Yeah. Also remember that after the session finishes at 3, we'll arrange for coffee by 3:15. You are then free to do whatever you want, go out into town, travel around, wander around. When you come back, dinner will not be in the place where it usually is, which is upstairs, but have dinner with the regular, in the regular canteen, which is on the ground floor. And you can tell them that you're a participant and they'll write your name down, or tell them what your room number is if you're staying in the guest house. Okay? So it's otherwise a great pleasure to have Professor Nagasuma Chandra from the Department of Biochemistry at the Indian Institute of Science. Suma has a really punishing schedule today. She came in the morning, she will give 2 lectures and then she will leave soon after. So you should catch her in the coffee session if you have questions for her. And Suma, go ahead.
Thanks very much, Gautam and Sitabra, for this kind invitation. I'm actually not a mathematician, but we do use a lot of modeling to understand biological processes. And in this case, I'm going to tell you a little bit about how host pathogen interactions can be modeled, different approaches that are there, some case studies to kind of give you an overview of what has been done in this field. Of course, there are many open questions, and I think it's a ripe time to be looking at many different approaches. Somehow there's a lot of data available, the methods available. Of course, there's no dearth for computational power. So I think it's just a matter of putting things together and doing it. And also a large number of high throughput methods for validation are available, which is making it a very exciting time to be working in this area. Okay. So let me start with a little bit about what we understand about infectious disease. Okay. So the first question we want to address is what makes a bacteria or a virus pathogenic, right? So in fact, the word pathogen cannot actually be defined without the context of the host, because the same virus or the same bacterium of the same strain can be a pathogen for an individual at a given point in time, and for the same individual it will not be a pathogen at another point in time. I'm sure you all encountered this at various times. You don't come down with the flu every single time you're exposed to the flu virus, right? You don't come down with some other infection or a wound or something each time you're encountering it. Okay, the same thing goes for 2 individuals. So one individual could be sitting next to each other and exposed to the same thing at the same time, but one person comes down with the disease, another person is infected but does not come down. So I want to bring in the distinction between infection and disease. Infection to us is just an exposure to the pathogen where the pathogen gains entry into the host cells, so that is infection. If the infection is not cleared and it manifests into a phenotype where the disease is felt, then we call it as the disease. So quite often, in fact most often, we do not come down with the disease. Okay, so most often the infection is cleared due to various host responses that occur in the host. There can be 3 outcomes basically. The first one is actual disease. In some situations, small number of cases it actually manifests into disease, where the pathogen thrives and proliferates in whatever niches it finds. The second outcome, which is the most predominant, is the virus or the bacteria is cleared from the system. So there's killing that's happening again due to host responses. And there's a third type of outcome, which is a stalemate, which means the pathogen is not killed, but it can survive within the host cells, somewhat in a dormant state or a latent state. And in some situations, such as what I'll show you a little later on, in Mycobacterium tuberculosis is a classical example of that. There are latent infections which the bacteria can reactivate even after decades. In fact, it's believed that something like more than 90 or 95% of the world has come into contact, especially in this part of the world, come into contact with the bacterium, and therefore we could be carriers without any symptomatic disease. Not that we have tuberculosis, but then the pathogen may be residing somewhere. And in situations of immunocompromisation, such as diabetes or any other kind of autoimmune or some other kind of disorder, or even HIV and so on, so the pathogen can become opportunistic come out, wake up from its dormant state and then cause infection. So these are the problems that we actually face with. So infection versus disease. Does infection with a pathogen always lead to disease? As I've just told you, of course it does not. Again, we might like to ask a question, why are some people more prone to getting it? Some people keep repeatedly getting an infection and some don't. So what is the difference between these, although the genetic makeup by itself may not be the thing? So it is of course host heterogeneity, could be related to some allelic changes or SNPs and so on, which we do not understand right now. While I am posing this question, I don't claim to have an answer, but these are the open questions that are still there which we could be looking at. In some cases, of course, we do understand link, genetic composition of some kind to susceptibility, okay, which I hope to talk about in Monday's lecture with a case study of influenza. Next, what processes or set of molecules in the pathogen are critical for its virulence, right? And again, take a bacteria such as Mycobacterium or E. coli, it can have something like 4,000 to 5,000 different proteins. Not all of them are important for its survival, and not many of them are required. A very few number of them are required for its virulence really. So survival is again distinct from the virulence. It can survive, but it need not cause disease. So only when it can survive, survival of course is a prerequisite for virulence, but if virulence factors are triggered in some form, then it can lead to disease. Now these virulence factors, there is not one molecule or two molecules, but there are a whole series of molecules, protein molecules and related sugars and lipids we understand much less about actually, but nevertheless some protein molecules that carry them, something is known about them. And this can be different from the same strain in different individuals. Okay, the route of entry can be very different, which is what makes it very complex. Okay, and our main goal in all of this is obviously to understand what's the optimal approach for first of all detecting it, that is diagnosing, then treating, and if possible preventing with a vaccine. So these are the three branches of medicine which are relevant to any infectious disease. Okay, so that's what we want to understand through this approach. Okay. Now again, why systems? This is more relevant to a biology audience. I think most of you here already have had some exposure to why we need to look at a much larger picture than individual molecules. Anyway, so traditional biological research, excuse me. So traditionally we've been looking at things with a reductionist approach, not because we didn't understand the the philosophy of systems, so to say, but because the technology that was available restricted us in some form to understanding the role of individual molecules, which has of course been extremely important. Without understanding the function of each molecule, you can't actually put them together into a system as a whole. But now, as I said earlier, sorry, it's somewhat a ripe time to be able to integrate these pieces of reductionist information that is out there and then see what are the interactions between these molecules that are available there, which protein is influenced by another protein. Are there, for example, alternate pathways? If you inhibit one molecule which is a virulence factor in a pathogen, that bacteria is still able to cause disease, it is still able to infect. The reason for that, when something is shut down, another route may open up, which may not be there in the initial stage and so on. So there are many mechanisms of that kind which are there. There are also mechanisms of how it interacts with the host. Bacteria are constantly changing, for example, and so are viruses. They change their cell surface properties, they change their antigenicity. So they are able to infect, but they change some molecules or some single amino acids in the right proteins, so that the immune system or the host responses can't recognize them basically. But having said that, the host also has its own armory of defense systems. For every move a pathogen makes, there may be a counter move that the host can make, and vice versa. For every move that the host can make, pathogen will find another method. If not in the generation of that particular individual bacterium, within the next couple of generations it will evolve resistance to drugs or resistance to the immune systems, and then therefore vaccines do not work very easily, especially for viruses where recombination rates are very high. Even for bacteria, unless unless you get the right knowledge about which is the target either for the drug or for the vaccine, it doesn't work because of this reason. They're constantly changing their landscapes that is visible to each other and so on. Well, host doesn't change its landscape like that, but then there is subversion systems that happen within the host. Okay. So, host pathogen together must be considered, and this is why systems become very important, and identification of interactions between components. We need to start at that level to be able to get a bigger picture. Of course, there are many methods of doing this, which I will talk about in a moment. And what is also exciting today is we at least have the starting alphabet. We have the complete genome sequences of these various pathogens that we are interested in, of several of them. Together with breakthroughs in proteomics, we are able to get measurements of RNA, of protein molecules, and also of metabolomes. In a number of cases, we have abundances of each individual molecule and how that varies over a time series in some cases, and how that varies in different conditions, and how that varies in different states of the pathogen. For example, what happens in Mycobacterium tuberculosis when it is actively replicating in a test tube, when it is actively replicating in the context of a host cell. We have models now, cells, macrophage cell lines, where we can actually infect Mycobacterium and then see what are the changes that happen within this bacterium. And what happens when it is in a latent state? These are the various stresses, nutrition stress, oxygen stress, pH stress, and so on. We can get all these kind of measurements in place. I mean, it's not very easy or trivial to do this, but then it is feasible certainly. Okay. Okay. So again, the ultimate goal we have already talked about. So let me just move on to the next one. So what are the different known routes or known goals of how a pathogen interacts with the host? First and foremost, it needs to gain entry into the host cell, right? So the host receptor is important, and depending on which host receptor it is able to attach to, the tissue tropism can change, and the outcome of the disease can change. This is well known. But again, it may not actually enter through one particular receptor. It may be multiple receptors at the same time with different quantitative profiles. And this is why again systems become very important. An example that I have given here is again for Mycobacteria. See, there are, I do it the right way. Yeah, here you go. Mycobacteria ligands. We have here opsonized, coated, coated mycobacterial molecules. We can have antigen 85C and lipoarabinomannan. There's a complex carbohydrate. Basically, these are the ligands from the mycobacterial side. Then we have manlam, again a complex carbohydrate, and there are many other unknown, of course, yet to be characterized very well. And the counterparts, the host receptors that are well known, are Fc gamma receptor, which is very important for phagocytosis and so on. We have a complement receptor 3, important for triggering the innate immune responses. Again, mannose receptors, scavenger receptors, toll-like receptors, all of these are important for immune responses, which I put on at a different level. Okay. Then it might also want to gain nutrition from the host. If it is a virus, it is of course utilizing a large amount of host machinery to replicate and so on. But even in a bacterium which is reasonably self-contained like Mycobacterium, in many situations it is hijacking the host machinery to deprive or deplete resources in the host cell and utilize it for its own benefit. Like a classic example again is iron. Iron is a very essential nutrient for all of us. Okay. In a situation like infection with Mycobacterium, there is increased utilization of iron from the host cells into the mycobacterial cell, and mycobacteria also require iron. So there is actually a competition between both of them. Again, there is a model which I will come to a little later on. In fact, there is a concept that has come out called nutritional immunity here. So tuberculosis patients, any patients for that matter, are fed with extra vitamins and iron and mineral supplements. It has been observed in the clinic that more the iron that is given, more the chances of tuberculosis proliferating basically. And this was a kind of a dilemma because it didn't make any sense. Then later they found out that it was indeed, the bacteria was getting more nutrition by feeding the individual with more iron. So which was really counterintuitive, but that's what actually happens. But having said that, it doesn't happen all the time in all individuals. Again, there's a lot of heterogeneity, but there is a significant amount of significant number of people where it actually happens. And coupled with the fact that tuberculosis is seen more in people with malnutrition or poorly fed states. This becomes a very very big problem, in fact. Okay. So another kind of fuel or that it utilizes is cholesterol. Cholesterol as a carbon source. It doesn't really care for whether there is a carbohydrate like glucose or citrate or whatever. It can utilize the fatty acids, degrade them into some form, convert it back into carbon resource for itself, and then do that. So there is plenty of cholesterol and ligands of that kind which can get converted into cholesterol in the host cell. So that is again another big challenge, and not very much is known about it. Of course, a large number of papers looking at individual molecules, but the system as a whole is yet to be looked at in great detail. There can be things like isocitrate lyase, a particular enzyme which is like a shunt. It utilizes to the carbon source via the glyoxylate shunt pathway. The citric acid cycle that most of you may be familiar with does not go through in its full cycle. There is a bypass somewhere, and the reason for that is an enzyme present in the mycobacteria called isocitrate lyase, which will bypass several enzymes and go through that. So if you believe that this particular enzyme that is there in the citric acid cycle is a good drug target and you inhibit it, the shunt becomes very active and it goes through that particular thing. There are other things such as LPD and SUCB, various protein molecules play critical roles in the synthesis of acetyl coenzyme A, again a very very important cofactor for all central metabolism out there, okay. Also important for the glyoxylate shunt. Host on the other hand, what does host do? There is this IFN gamma, interferon gamma, an important cytokine, and tumor necrosis factor alpha, another important cytokine. This down regulates the TFR and ferritin. These are the proteins that are important for iron acquisition from each other. So transferrin and ferritin expression is down regulated in the host upon infection. How exactly that happens is still a mystery. I mean, everything must boil down to thermodynamics and some quantitative numbers of molecular recognition and the population of ligand and population of receptors that are out there, as well as the affinity between the two. That is yet to be known in many cases, but small examples again we know quite a bit. Okay. So IFN gamma inhibits all other sources of carbon by excluding Mycobacterium phagosome from recycling endosomes. So both are actually doing something. Which one wins will again be a number game, which one is more in number or more active, and so on and so forth. So the next kind of level of interaction is at the virulence level. So virulence factors, some examples of virulence factors are the cell wall itself. In fact, in mycobacteria, the cell wall is so very different from other bacteria, which is why it is difficult to treat tuberculosis with regular antibiotics. We get streptococcus, staphylococcus, all sorts of strep throat, all sorts of infections that we commonly encounter. We take a broad spectrum antibiotic, we don't go to the clinic, the doctor can't take a swab and then do a culture and then find out what the pathogen is and then find out its sensitivity and then treat us. It will be too long before we are given the treatment. So instead a broad spectrum antibiotic is given which works against a large number of gram positive, gram negative or both kind of bacteria. But mycobacteria is very different from that, and so are other hosts. I am just using this as an example, but it applies to many other situations. This is different because its cell wall is coated with lipids of various kinds. And one such example of the one major constituent of that lipid rich cell wall is mycolic acids, which are about 60 carbons in each molecule. They are unsaturated. There are varieties of mycolic acids, and this is like a warm fatty coat for the mycobacterium, which makes it impenetrable for antibiotics. Antibiotics cannot enter because of this particular thing. Okay. So, and the other one is cellular metabolism. Some metabolism is altered. It is able to use like fatty acids. That is one example actually. So, an example of the cell wall, I have said these are the other constituents of the cell wall. Lipoarabinomannan. So, we have PD, IM, PCA, various lipid molecules these are, okay, trehalose and so on and so forth. And then metabolism mainly for fatty acid synthesis. And then there are these stress response proteins, heat shock protein and various other stress response proteins. For example, there is a nitrate reductase in that one which is able to counter oxidative stress much better than many other bacteria can do. Iron uptake I have talked about. There is also other uptake, manganese uptake and magnesium uptake and so on, and a variety of these which I won't go into detail for that. Again, the two other major things is what are the critical host cell pathways that are important for responding to these things. So, the host cells for enabling an infection, or this is there for a variety of reasons in the host, because we are constantly encountering various foreign organisms. We need to be able to take them in, kill them or evade them in some form and throw them away. So, that we are evolved with these systems in place. The phagosome maturation, phagosome means it is eating up this particular pathogen, and there are signaling pathways. We have a calcium signaling, PI3K signaling, P38, ERK, various signaling pathways that leads to cell killing of the pathogen ultimately. And on the other hand, we have immune evasion levels. Immune evasions can come from reactive nitrogen intermediates, reactive oxygen intermediates, or MHC class 2 antigen processing. It can fool the, pathogen can mimic some of the host molecules and fool the immune system by making it believe that it is actually a host protein or host molecule and not a foreign molecule, and therefore immune system is shut off. If our immune systems work against our own molecules, we will have autoimmunity. That should not ever happen. And so these change their flavor, either the amino acid signature or the carbohydrates or whatever, so that it looks like the host molecule itself, and that is what actually happens. Or it can, variety of fusion events like phagosome, lysosome, fusion events can be subverted. We can have various things that the host does and various things that the pathogen also does. So, our modeling strategy, we want to obviously look at everything, but modeling strategy will depend on what specific question we are asking at that time, which is not new. I am sure it applies anywhere for any modeling study. So, the things that are important is what is your actual question, what are the answers that you are looking for, at what level, and what data do you have available to you. So, these are the two things that will decide what sort of modeling strategy you can use. If your question is, for example, to understand the growth kinetics of a particular bacterium in a host environment, then it is sufficient and also efficient to look at it at a very gross level. So, you just want to model the entire thing by a couple of equations basically. So, you can look at ODE like models for that, and that will suffice, because that will give you the information that you are looking for, and you may be able to get quantitative rate parameters of various kinds from experimental data that is available, fit it, and then ask new questions on that. But if your question is to understand which particular molecule is acting as a bypass mechanism in this particular condition, then those models are not usually adequate. You might require a more detailed model. In fact, what I show you here is a taxonomy of models that is applicable in any area of systems biology, not restricted to host pathogen interactions alone. So, we have this low granularity models where the detail is lower, but the scale is usually much larger. High granularity models which are very detailed, but it is obvious, for obvious reasons, it is restricted to a smaller number of components basically. So, at the one end of the spectrum we have atomistic models, which could be molecular recognition or intermolecular energies, for example, or even molecular states of a particular protein, conformational states of a given protein, and so on. So, these at this end we have that. Then if you, as you go up in this ladder, we have differential equations where you are not actually looking at individual atoms of individual molecule, but you are modeling a whole, obviously differential equations can go at any level, but if you are looking at, for example, biochemical reactions, enzyme A is catalyzing a reaction of A to B and so on and so forth, you could easily model them here with if some parameters are available. I will go through some examples as we go along. You can of course put in stochastic parameters when you do not have, and there are uncertain processes and it is not very clear, and many possibilities you can incorporate them again anywhere. The next level of model, again typically used for metabolic systems, biochemical pathways and so on, is something called a flux balance analysis, again borrowed quite heavily from chemical engineering and fermentation industry kind of areas. So, I will describe this as well in a little bit of detail. Somewhere I am not really sure if it should be in this hierarchy or not, but approximately in this hierarchy there is Boolean modeling somewhere. So, you do not have very good quantitative data, but you have some sort of binary data, yes or no, and then you have Boolean rules. So, you could easily utilize Boolean modeling for that. I do have a case study for this. Of course, topological analysis using networks, like road networks of a city, that are again possible for a large scale when quantitative parameters are not available, but some other type of data is available, which I will show you. You have somewhere in between machine learning networks and so on. If you have a large number of genotypes and a large number of phenotypes and you do not know anything in between, you can actually train a system and then get some correlations and use that for prediction for any unknown situation. So, machine learning again works for a variety of situations. It can very well be used for host pathogen interactions also. I do not have case studies here, but it is easy to envisage a situation where you have multiple strains of a given virus. In fact, if you look up repositories that are available for say influenza virus, one particular type A, H1N1, one particular thing, you will easily get something like 1,000 genomes, 1,000 different strains, so 1,000 genome sequences are available. And clinical evidence is also available for which one has caused an epidemic and which one has not. So, simply with this information, you can train, you can use a machine learning approach, train either neural networks or support vector machines or a random forest or one of those, and then see that which particular strain and which feature in this particular strain may be more related or correlated with the phenotype that you are looking at. You can have a two-class classification where disease, no disease, or you can also have a multi-class classification where you have a gradation of the disease. So, all that is possible. The problem with those things, the advantage is of course you do not need any other data, you just need this starting point and the end point and you can get correlations. But the disadvantage is it is a black box. Many times you can't really tell what is important and whether a combination is important. So, it's of course, while I say this, there are number of advances that are happening in each method, each technique where they are trying to overcome the disadvantage that is out there. So, it may not be relevant in all cases, but by and large this is the disadvantage of machine learning approaches. Okay. So, you can also have logical models, which is what biologists always do, but maybe in a smaller scale. When you are trying to do that in a large scale, in a formal manner, you can call them as logical models or qualitative models basically. Okay, so how do we actually begin and what do we want to do? Again, I said it depends on the question. So, let us look at some more examples of where this kind of models are useful. So, you can use both a top down approach where typically this is in the realm of theoretical biology, which many of you have may be familiar with, which come down in this fashion. And today, because of the the data that is available, you can also do a bottom up approach and meet somewhere and then go of course both ways, that is quite possible today. So, let us begin with the bottom up approach. You start with the molecular components, and what data do we have for the molecular components? We obviously have the genome sequence that is available to us because that is there now for many different types of pathogens. So, the kind of models that we are looking at here is a sequence to function models. Sequence, it is now a linear string for you. So, you can use text matching methods, string matching methods of course by adding evolutionary information in various things, and you can have a sequence to function model. If I have this particular gene sequence or amino acid sequence, can I give it a name? Can I annotate it, right, just by looking at the sequence? Again, you can't do it by first principles. There is some sort of training that is happening. So, informatics comes into picture. This is typically the area of bioinformatics. So, you go and look at databases because have very sensitive methods to compare this sequence to all known sequences out there, and then say is it over the threshold, is the similarity over a certain threshold, and therefore, can you say they are evolutionarily related in some sense, are they homologs, do they have common ancestry, and therefore, is the function the same or not. Okay. So, that is the first level. You can also add in more information and say it is not sufficient for me to say there is 50% similarity or 60% similarity or whatever number that I give, but I also want telltale characteristics that are particular of a given family of protein. Take something like a kinase for example. Kinase binds to ATP, right. So, we need a signature and it is a motif within this large sequence that is known to be related to ATP binding in some sense. We also have signatures for a particular structural fold of that particular family. So, is that fold, is that signature present? Signature is somewhat like describing an elephant purely by its trunk. That is a telltale characteristic. If you say this is a large animal and it has a trunk, very quickly you narrow down onto very few and the top hit may be an elephant. In a similar way, you can look at motifs which are characteristic of that particular function. Okay. So that is the sequence to function models. And what sort of questions can you ask on that? If there is a mutation, a sequence is known, a particular bacterial strain, again I told you many strains are sequenced. In that protein, there is a change in one amino acid position or 3, 4 amino acid positions and so on. So there is a mutation. If there is a mutation, what happens to the function of the protein can be analyzed to a fairly confident extent depending on the data that's available, and then get some information. And if that particular protein happens to be the one that is interacting with the host, it's even better. You can try and understand whether it's more virulent or less virulent and so on. So we can have mutation studies, gene association studies. You can do the same for the host. Analysis of what we call as SNPs or single nucleotide polymorphisms. Between any two individuals, there are a large number of SNPs. Some are correlated with disease and some are not. Okay. In fact, there's a whole area of work that has come about in the last few years called genome-wide association studies. Again, a large correlative study by looking at a large number of patients and related healthy individuals, sometimes families of patients and so on and so forth, and trying to find out if a particular polymorphism is more linked to a given disease and so on and so forth. A lot of statistical filters are put in place to get the correct one against the large amount of noise that is generated from these things. So that is where it is. Whatever we can do with sequences, we can do it much better at a higher resolution with much more insight when we consider three-dimensional structures of those protein molecules. Okay, so those are the structure to function models basically. But then we are limited by the number of structures that are available to us. Why are structures more important? Because sequences, we can say a motif is present. We can say these two sequences are homologous to each other and infer the function in them. But when you look at structures, to some extent it is somewhat closer to deriving the function from first principles, because you can look at the three-dimensional arrangement of these important amino acid residues that are out there. And then you have a question? No.
First of all, when you look at structures, you crystallize, you keep it in a different environment from what it's a native environment. The second is often, I guess, 50% plus of proteins have large unstructured domains inside them, which may be critical in some way to their function. So, I mean, I guess the standard thing to say is that structure uniquely determines function and structural changes determine function. But how strong is this relationship thought of now, given what we now know about the flexibility of protein structures?
Okay, no, that's a very valid question. But in a large number of cases, we do find a very strong correlation. We are able to predict changes in function by looking at changes in structure. And we are also able to predict changes in structure by looking at sequence changes. So that where a good template is available, because our optimization techniques have improved to such an extent that you can actually get a good model. And if you take something, we take a human cell, it codes for about 19,000 plus proteins. For 16,000 or more, we have at least some part of the domain, some domain or something where a structure can be modeled. And for the Mycobacterium tuberculosis, we ourselves have done modeling for more than 70% of the entire genome. So which is not bad at all, by today's standards. Of course, crystal structures are limited. And to answer your first question, we have crystal structures in different environments, different space groups, for example, different pH conditions and so on and so forth. And the structure hasn't changed, which means it's not an artifact. Okay, we also have situations where the NMR structures and crystal structures are matching very well. So it's not really an artifact of crystallization, so on. And if you mutate some residues, the hypothesis that you've gained from crystal structures, you test it by mutation studies and so on, and it's worked out pretty well. So that way I think we're fairly confident of that. There are of course unstructured regions which get folded in vivo but not in vitro. There are many such situations. Yeah, right. So this is where we are. So the next level of hierarchy, if we go up here, again in some steps, okay, you can actually fill it up. It will be a continuum of sorts, but just for convenience I have put it in this fashion. So you have metabolic pathways and biochemical reactions out here. So what sort of modeling can we do? We can do it both for the host. We can also do it much more easily, obviously, for the pathogen. So an example here would be to model a single metabolic pathway. Like I talked about mycolic acids, we can actually model the entire mycolic acid biosynthetic pathway, and we can model fatty acid pathway in the host cells using kinetic modeling. These are ordinary differential equations. You have rate kinetics. Basically, you have some assumptions that they follow Michaelis-Menten kinetics or even allosteric regulation you can put in place. And I am assuming that most of you are familiar with it. If not, please feel free to ask, and I am happy to explain that in more detail. So, you can look at if this is the starting concentration of the starting compound that gets into the pathway. A pathway is now A to B to C to D to E metabolites, each one catalyzed by a given enzyme, A to B by E1, B to C by E2 and E3. It can have more than one enzyme as well. This is a sequence of biochemical steps that go on. So, if you have velocities of each enzymatic reaction, Vmax values, which are available for many things, especially for the host system, then you can calculate if a given compound, we'll take glycolysis for example, if you have glucose as a starting point, how much of pyruvate is made at the end of it. That is the end product of glycolysis. So, this much glucose, these are the intermediate steps, and this is what is made. And if you perturb one protein, such as mutation, where the velocity is changed or increased or decreased, or the affinities for these substrates are changed, they bind these substrates poorly or more strongly or so on, then you can predict what happens to the system. What are the intermediate fluxes and what is the final amount of product that are out there. So, that can be modeled quite easily with ODE models. But in a number of situations, we do not have these Vmax values that are available to us, right. We know there are cars on the road, we do not know how fast it is going at a given point in time. Your Vmax is the maximum velocity at which an enzyme is functioning basically with the given concentration of the enzyme. So, that is not available, but however, we know that A becomes B and B becomes C, and we also know stoichiometric coefficients. We know we need 2 of A for that to become 1 of B and so on and so forth. Like to give you an example, 1 molecule of glucose becomes 2 molecules of pyruvate eventually, because a 6 carbon sugar becomes 3 carbon sugar. So, you will get 2 of those. So, we have stoichiometric coefficients that are known from some species or the other, and these are very well conserved throughout evolution. You do not see too many changes that are happening. You do see changes occasionally, but not all the time. So, that is available. So, there is a technique called flux balance analysis, which I will describe in later again. So, we can use constraint based methods for that when we do not have information which is very quantitative, but we have somewhat semi quantitative information, we can put them together and do a modeling of this kind. So, in some situations we do not have even this information, we do not know the function of this protein at all, but we may know what it interacts with. Again, I will come to that in a minute. So, we know somewhat protein-protein interactions. In the sense, it is now very similar to a road network in the city. Right? We do not know the function of each junction, but we know which junction is connected to which and how the flow happens in some sense, or what are the possible flows that are happening. So, that we can map it as a simple network, and various methods to capture host networks, pathogen networks separately, and host pathogen networks also. Again, I will explain that in detail. And Boolean models also I will come to. So, these are intercellular influences can be captured. The beauty of Boolean models is that you can use any level of hierarchy and combine them in a single network of a kind. You can do that. And from the top down, or you go, the cell is in between point out here, because it is a reasonably self-contained unit where you can model various things. And you can have here multi-scale responses such as spatio-temporal heterogeneity, tissue formation, and cancer metastasis for example, or how an infection spreads from one organ to another. All of these can be captured in here. A little further you can have things like host pathogen coevolution, which I think for lack of time I will not go into that today, but there are just a handful of papers where people have looked at game theory to say how a pathogen can evolve. Okay. What changes can happen in its genome and whether it is useful for it, or what sort of things can be modeled basically. Again that is a very interesting area to get into. And at the end of the hierarchy we have disease epidemiology. You can look at population dynamics of disease spread and so on and so forth. Okay. So, choice of modeling strategies, as I said, only a portion of them put down here, will depend on whether you want to look at a discrete state or a continuous state, discrete time scale or a continuous time scale. Of course, you have here when the state is discrete and the time is discrete, you can have agent based models and Boolean networks. When it is continuous, you can model them more as Petri nets and so on. On the other hand, we have here difference equations, rule based models, or when both are continuous as differential equations of kind. Okay. I I'm sure many other speakers have spoken about ODEs, and I'm not an expert in this, so I'm not getting into it in detail, but just for completion, I have kept a slide. I must also tell you there's a lot of text in the slides from now on, so I may not read out all of this, but I thought it may be useful for the participants to take it back and then look at it. It's at least reference material. So do bear with me when you see a lot of text in the slides. Okay. So here these models are generally deterministic in nature, and then you can, like I said, very useful thing would be to look at biochemical reaction, signaling cascades that are out there, or even larger processes such as cell-cell adhesion and how entire pathogen, rate of entry into the host, for example, depends on the parameters that are measurable experimentally and what can be modeled. So and usually it is the number of components here are not very many for obvious reasons. You can't really solve very large systems. So about 20 to 30 would be fairly largish. You have many models which lesser number of components out there. Let me skip this as well. Just a quick example here of continuous time, continuous state model, well represented using differential equations. So this is an activatory and inhibitory interactions between 4 species A, B, C and D are shown. So these are the set of differential equations where the update of one variable is dependent on other variables, which in turn is time varying and dependent on other set of variables, which are therefore become coupled differential equations. And you have a plot which shows A in blue coming down, B again going up, C going up and D also going up with this model out there. A gives rise to B, D also gives rise to B, but B becomes C, and so this is in itself is reasonably complex, although there are only 4 components. Okay. Again, for an infection situation, you have somewhat like this. You have a resting macrophage, and you have an infected macrophage when it encounters a bacterium. An infected macrophage can get further infected with another bacterium when it encounters. And there is a maximal carrying capacity C that we use somewhere. So the number of bacteria within a macrophage must be within that limit. And again, you can model them with the equations that I think again most of you may be familiar with. So let me skip that. There will be a burst rate, there will be a birth rate for a macrophage, there will be a birth rate for the bacterium, and there will be a death rate as well, which is a normal thing. So you can take all those factors in and then get a model of how many macrophages are infected with different number of bacteria. So,
Suma, where is that model from?
This one?
Yeah.
I think these are all from Kirshner's papers. Dennis Kirshner. She has a massive review of mycobacteria. So, Okay. Again, PDEs, partial differential equations and compartment models, which I'm again not an expert in this area, but given the complexity and heterogeneity in the host system, it's often difficult to place them in a single differential framework. So in order to address the temporal and spatial evolution together, some multi-compartment models and partial differential equation based models are employed. There are many examples. This is there for, say, modeling blood flow. This is there for many different types of situations which are used in biology. Obviously, they can be used in host pathogen modeling as well. So the entire space can be subdivided into well-mixed smaller compartments. I'm sure Gautam here can talk about this much more, and each of which has its own governing equations, while communication among them is allowed through interface equations and so on. So the next type of method is agent-based modeling, which is gaining a lot of popularity, and there are a number of different ways of implementing this as well. So this is a mathematical formalism that can be applied to address the complexity of biological systems in a computationally tractable manner. So that's the advantage of that. It's a generalization of what people call as cellular automata, systems that enable simulations that are discrete in time, space and state. So you can actually do that. So these involve what autonomous entities called agents. Each agent can talk to other agents based on a set of rules that you define. They have defined spatial attributes. You can have agents with a tag of a spatial location at a given point in time, and you can have, for example, in a host cell you can have agents across compartments. You can have an endosome, you can have a phagosome, mitochondria, Golgi, all of these various compartments you can have, and bacteria itself can be another agent out there. You can also have different proteins in the bacteria as different agents as well. So there is no problem in mixing up hierarchies in these things, as long as you can define the rule reasonably clearly. So predefined rules dictate the interaction among agents and their environment. Now these you can capture stochasticity and heterogeneity in systems quite easily, contrary to the regular ODEs. So these models exhibit modularity in structure, enabling independent modifications in rules. So you can make a modification in a rule, you can evolve the models quite easily, is the take home from this. Now states of agents are updated in an asynchronous manner. You can have again both asynchronous and synchronous depending on what sort of rules you want to make, and you can allow autonomous and adaptive decisions at each agent. Each agent can be performing a multitude of tasks and give a final decision which is transmitted to the next agent basically. So, so you can have asynchronous rule firing. Agent models are much more versatile as compared to cellular automata and are capable of emulating interactions of living entities in a more accurate manner. So again the take home is that you can model biological systems much more realistically by this formalism. So it's just a useful tool for us to do that. So in an agent based model, the interactions on a local scale can transmit to an effect in the entire system. So that's the beauty of it. You can have local interactions because agents talk to other agents and there is some sort of a flow that is happening. You can see the effect that is more widespread actually. So they are particularly useful for studying complex systems in which individual heterogeneity in spatial interactions are important. Okay. So another kind of thing, Petrinate sometimes used kind of synonymously with other methods we have seen, especially ABMs. These are basically graphical representations of interactions consisting of two types of nodes, places and transitions. Okay. So nodes of different types are connected by direct edges. I'll show you a picture in a minute, called arcs, while no arcs exist between places or between transitions, somewhat like a tripartite network, you can think about it like that. A standard Petrinate model comprises of 4 primary components, which are places or nodes that represent local states or system components. Tokens are marker variables used to indicate which nodes can undergo transitions. Right? If a token is of a particular kind is present, a node can change its state. It can change conformation, for example, or it can get a T cell can become an activated T cell. These are the examples out there, and at a given time step. And the set of nodes with tokens for a given transition are updated synchronously, and tokens are passed on to neighboring nodes in the transition arcs based on the predefined set of rules. Again, there are rules of which can transmit to which one and when and upon what trigger. That kind of thing can be updated here. So transitions capture rules to dictate state updates. State update is now what is the change in state of a particular component out there. Like again, as I said, an immune cell becoming an activated immune cell upon receiving a ligand. An antigen, for example, binds to an antigen presenting cell, and that recruits a T cell by binding to the T cell receptor, and then it becomes activated. So that kind of a change of state can be captured in these things. And directed arcs are the routes through which these things can be transmitted. So you may see something in common in all of these kind of things that I am talking about. There are nodes and there are rules of how the nodes themselves change and how they transmit the thing. So what is different in all of these is how much flexibility it allows and how much complexity it allows, and what is the formalism and its computational ease basically. Otherwise, conceptually they are seeking the same kind of answers. An example of an agent based here. So somewhat like a grid, you can have a multi-dimensional grid where you have a location for each individual agent. Therefore, you can tag what happens to that and so on and so forth. As you can imagine, this is very useful for tissue remodeling kind of thing, to understand what happens to a given tissue upon say infection and whether an infection is moving. Let us say if Mycobacterium again, it is moving from the primary site, which is the lung macrophages, into the generic circulation. It can get into spleen, it can get into brain, it can get into various kinds of things. So this kind of a thing is useful, although I am not familiar with anybody having used this for Mycobacterium at least. Okay. So these are the tokens and transitions and places, and I think you've got a good idea of these things. So now again, this is for a simulation for infection situation. You have a resting macrophage, infected macrophage, bursting macrophage, same situation. the same model of phagocytosis. So you can use, I won't read out the whole thing, but you can define contact with a distance threshold D, how far should that be, which is actually capturing is a molecule on the pathogen interacting, making a, is it recognized physically by a molecule on the host cell. So in what range of interaction is it present? So it's contact kind of a thing, which depends on the time and how fast it is allowed to move within the cell and so on and so forth. So external bacteria in this case are free to move upon contact with macrophages. They get internalized with a probability value Pn, after which they are marked as internal bacteria. So a bacteria can be outside the cell, with some probability it is allowed to interact, and then it becomes an internalized bacteria. And this has no free mobility, so its states change, the state of the bacteria changes, and instead it is it moves along with the macrophage that has ingested it. But these bacteria, macrophages are meant to kill these bacteria, but in situations like Mycobacterium and Salmonella and several other intracellular infections, the bacteria are clever enough to evade this killing process within the cell. Instead, they use the resources from the macrophage and thrive inside, which is a bit of a paradox. Okay. So this is what happens when the internal bacteria count exceeds the infected macrophage's carrying capacity, the latter bursts. So again, it typically a given macrophage cell can carry up to 6 individual bacteria of Mycobacteria. It will be different for different bacteria. So what it's not to size, obviously, bacteria are much smaller than this. So when it exceeds that, a cell can no longer hold that thing, so it bursts. And these bacteria gets into the circulation, can either get killed or infect new cells. Both can happen, and depending on the numbers and depending on the immune status, in Mycobacteria, a granuloma can be formed, a large number of other cells can get infected. It's a similar situation with viruses, actually, that's what actually happens. So though the model proposed here is highly simplified, the strategy can be used to extend the model to simulate real systems. So there's a lot of extension that's possible in all of them. Okay. Again, I think I've touched upon this spatio-temporal thing, let me skip. We'll go on to Boolean modeling basically. So Boolean modeling now, just as the word says, from on, or basically started from the mathematician Boole. So Boolean network models, which were initially proposed as prototypical models of genetic regulatory networks. Now I don't know how many of you are familiar with genetic regulatory networks. Do you want me to explain that, or are all of you familiar? You want, okay. So now we have a large number of genes, right, in each cell. Not all genes are switched on at a given point in time. Okay, switched on is the level of expression. How much is it expressed, which indirectly tells us how much of the protein is made. So the moment a gene is expressed by a certain amount, that much mRNA is available, and therefore that much protein can be made. That is what it is. And so these are dynamically controlled beautifully by a regulatory network. And most predominant components of this regulatory network are transcription factors. It's like a master switch. You may have a master transcription factor that switches on various other switches, and when you switch it on, various other molecules can get switched on. But unlike in the electrical systems that we quite often see, each light is switched on to a different extent. The intensity with which it glows can be different. So some are very dim, but some are very bright, depending on the requirement. We don't come across intelligent systems of that kind commonly, but I'm sure they are available already. But in the cells, these are really, really well regulated. So those are genetic regulatory networks. So Boolean modeling was initially started to capture this, which if a particular transcription factor is present, something else is either inhibited or activated, both can happen. Many are actually repressive kind of edges that you can draw out there. And the mechanisms are also quite well understood, how it binds to a particular region in the DNA and therefore it activates or represses, those are quite well understood. So it started from there. A Boolean network consists of a set of nodes whose state is binary. So it's either present or absent, typically 0 or 1. That is the state for each component in the Boolean network. And it's determined by other nodes in the network through Boolean functions. So as you can imagine from a gene regulatory network, if let us say a transcription factor A is expressed, then its target protein B is not present, so it becomes 0. And if B is not present, some other thing can become activated because B has a repressive nature. To give you an example again, kinases. If a kinase is not present, it doesn't phosphorylate something, therefore no signal is there, therefore the end molecule is active or inactive. Both ways are possible. So, you can have for the entire flow 1 0 1 0 or 0 1 kind of relations. Excuse me. I'm okay if you're all fine. I hope it's not an infection, but it's an allergic process which is just started. So okay. So after assembling individual components and regulatory interactions involved in a system into a coherent network. Now you put all of these together. Again, I'll come to an example which may be more easy to understand. It becomes a network if you have many rules which are interconnected with each other. That's the message out there. So you can model qualitative temporal behavior of the system and to understand how perturbations may alter its behaviors. That's the main thing, to make one model of a kind and then model perturbations which are either mimicking each individual patient or each individual bacterial strain and so on. Once you have a basic model, you can do a large number of these things. So they also lead to predictive testable hypotheses which are especially valuable in poorly understood large scale systems. I do have an example a little later on. So these have been successfully applied in modeling many gene regulatory and signaling networks as well in a variety of organisms. So in addition, they have been used as models in reverse engineering of biological networks. That is to find out what is the missing link between this regulatory pattern and that phenotype that you are looking at, and therefore whether another molecule should be involved or not involved. That kind of questions you can easily ask. Okay, an example is given here. So this is, I think, general introduction. We'll leave that. So a wealth of molecular level qualitative data on individual components and interactions can be obtained from experimental literature. That's already there for many systems, and high throughput technologies. Right? You can actually get like I said, microarrays or RNA-seq, which is a part of a next-gen sequencing kind of technology. You can measure which gene is expressed to how much in a given condition, and you can do that for all genes in a given system. So it's a high throughput technique which does that. We also have technologies like proteomics and metabolomics, which gives us abundances, so in a high throughput manner. So this is qualitative in nature. But then it's often sufficient to capture the topology of the network. It may be just 0 and 1, but it is sufficient to capture the topology and to understand which is influencing which. So some system dynamics come into picture here. And these Boolean logic functions or Boolean transfer functions are used for describing interactions between different components. So a system with n variables has 2 power n distinct states, because both are, it's just 2 states for each node. An example again is given here, same example of phagocytosis. Here we want to look at, there is a resting macrophage M engulfs extracellular bacteria BE and becomes an infected macrophage M1, okay, rendering extracellular bacteria as intracellular bacteria. Same example again with Boolean rules out here. So M1 is M and BE together becomes M1, and M1 or MI it should have been. So your BI is either BE and M or BE and M1. Both of them can give rise to M1 basically, infected macrophages, which is shown better here in this picture. So you have here 3 vertices in this network structure. This is projected as a directed graph where the node set vert V1, V2 to Vn, only 3 are shown here, corresponds to the Boolean variables, and the edge set E is implicitly defined by the Boolean functions of the model. Each edge set here, if you have E1, E2, E3, that is defined by the transfer functions out there. Example transfer function B1 with this sigma 1, sigma 3, it means that this or not this thing. So you can have A but not C, A and B both are required for this. Those kind of rules can easily be made. Just like how you combine various and, or, but not, and not kind of variables to do for example PubMed search. Right? You want to find exactly that paper, but which does not talk about a text search you can do for various kinds of things. But here you are trying to do that for understanding molecular states. Those are written out as each individual condition now is written out as truth tables. So if these are the variables sigma 1, 3, 1, whatever, you can write them as 0, 1, 0, 1 kind of rules. and what you get is a state transition graph. At the first state, 0, 1, 0 means 0, 1, 0. So, it is representing the entire system in zeros and ones in the order that is predefined. And then that can, in the next, it is updated periodically. Again, it can either be in an asynchronous fashion or synchronous fashion, depending on your transfer functions that is out there. And the next state, it becomes 101, but 101 can also become 110, and they are interchangeable. This can go back to this can go back to this, and so on. Or you can have state diagrams like this, 011 to 001 to 000 and so on, which is describing the state of each individual component in this thing. And the transitions are looked at, and then finally when it becomes 111, it can't become anything else, it can't change into something. So, it could be the end phenotype that you are looking at, for example. So, tracing through these transitions is a very interesting kind of a thing, and you can ask a variety of questions. You can also find out what is called as the attractor. That is, do a large number of given initial conditions end up in the same thing, whatever, and what are the routes through which it ends up in the same state. That is, I think, defined as the attractor basin and things like that. So, there are a number of ways by which people do that. So, a Boolean variable assumes two values, 0 and 1, corresponding to the logical values. Of course, recently there have been many advances to this methodology, and you do have what are called as more quantitative Boolean models or probabilistic Boolean models, where the value is not 0 or 1, but it can be anything in between, which means you can model a variety of in between situations also. But then you will have to define a threshold, whether 0.5 is your threshold or 0.8 is your threshold for the next act to be fired, next rule to be fired, is something that you will have to define. Okay. So, here the logical values are false and true basically. So, when representing the state of a biological entity by a binary variable in two states, these are off and on, this is what it is known as. So, a Boolean function with k variables is a mapping B, now 0, 1 with some probability going to 0, 1 from the set of all k tuples over 0, 1 basically to a binary output. And this function describes how to determine a Boolean valued output based on certain logical operators from k binary input. So, just a way of modeling this. I've already talked about this. So, the truth tables also we have seen. So, I can skip the rest of this slide. So, now there are two types of update schemes, synchronous in the sense all are updated at the same time based on their previous state. Whereas asynchronous, there is an order. If A changes, then A1 in the first state changes, and A to A1 if it is changed. B1 depends on A1 and not on B0, right. If A0 is going to A1 to A2, at B0 is going to B1 to B2, the B1 does not depend not only on B0, but also depends on A1, because A1 is feeding into B1. So, this is an asynchronous kind of update, and there the order becomes very, very important. So, you need to know which event is triggered after which one, which typically comes from biological knowledge. Okay. One can, if you do not know anything about that particular system, you can do variety of, you can do all possibilities basically, and then see which one is mimicking the real situation more closely, and then take that and use it for further predictions. So, and you can also introduce a delay term here, which is what we have done in an example I will show you. A delay can be that you fire this event only after some number of time steps, right. And a typical example that we have looked at is when the adaptive immune system is triggered upon an infection. Innate immune system comes into play immediately, but adaptive immune system is fired only after several cycles. So, you can put in the delay factor. Okay. So, the variables are updated in a non-synchronous manner. For some states or a set of states, the state remains stable, it does not change at all, and is known as an attractor. That is the state transition, I said it keeps coming back to the same thing, and it is a representation of a long-term system behavior. In a sense, this is the most stable state for the entire system, that is your combinations of 0 and 1s. This is the most stable or most probable outcome is what you can see. As the system depends on the initial conditions, changes in the initial state will obviously result in different attractors after simulation. So, one needs to have some sort of an input of whether an initial thing is on or off, and typically that is taken from experimental data, and for gene regulatory networks, modulating host pathogen interactions or whatever, you take it from expression data or proteome data typically. So this is a typical workflow for constructing a Boolean model. You start with the network structure of the model. If you are doing a large scale system, you start with literature, experimental data and net synthesis of some kind. And this is not, it's predicted to some extent, but it is also putting together all known data about a particular system. Then to write out the Boolean transfer function, which is a direct effect of knowing whatever you know here, and then getting the transition of system states. So you have to keep in mind the biological time scales that are relevant for this thing. If it is molecular recognition, you may be looking at microseconds kind of time scale of an individual event. If you are looking at cell division, let us say in a mammalian system, it can go up to 24 hours or longer. If it is a bacterial system, it will be usually half an hour to 1 hour to 2 hours, that kind of a time frame. So depends on what question you are asking. And analysis of the system output, obviously test the correctness of the model and look at biological implications and predictions. So at this stage you are getting biological initial conditions and some idea of the attractor of the system, and at this stage you are looking at whether the known attractors are consistent with what is known in literature. So you validate the hypothesis in some form, or if you can go back to the bench and then validate it experimentally, it's even better. So is it matching with known dynamic events, matching with known states of components and so on. Then once it becomes where you are fairly confident with the model, it becomes predictive, then you can look at long term behavior, look at roles of interactions and loops. Are there feedback, feed forward kind of inhibitions, are there cycles within this big thing so that are influencing each other and so on. And then models say knockout and overexpression such as heterogeneity between bacterial cells or heterogeneity in host cells, those kind of things you can capture out there. Yeah, again example here of people also use something called a piecewise linear model. So this is a directed network associated with the model which forms a negative feedback loop, only one portion of the Boolean model here. So the Boolean update functions are completely determined by this network. You have A which is activating B, B is activating C, and C is inhibiting A. That is the symbol for inhibition that is typically used out there. So C is inhibiting A out there. In fact, there is a classical study called repressilator, not for host pathogen interactions, of just transcriptional regulation. That is in fact started the area of synthetic biology. You might like to look up the repressilator studies basically by somebody at I think Leibler from Rockefeller. So it's a beautiful thing. So if A is activated, B is repressed. If B is activated, C is repressed. If C is activated, A is repressed, and so on. So it's in fact children play this kind of a game, and if one stands up, he or she hits the next person, hits in the sense makes them sit down, and so on and so forth. So then at a given point in time, who will be standing up and what is the pattern? And you will see oscillations of this kind if the time is the same between them. So you will see beautiful oscillatory patterns that go on. So the directed network associated with the model forms a negative feedback loop in this case. So the Boolean update functions are completely determined by this. The state transition graph of this is shown here. So you have 0, 1, 1 becoming 0, 0, 1, that's 0, 0, 0, and then this becomes 1, 0, 0 because of its inhibitory nature, and then it goes round 1, 1, 1 becomes 0, 1, 1, and so on and so forth. Okay. So it has two limit cycle attractors which are sustained oscillations, and the time course of this continuous variables associated with nodes. I don't think you can see the green very well. Anyway, green triangles, this is supposed to be green, blue circles and see red squares according to a piecewise linear model. So you take each time step and then get it and then put them together later on. Okay, more text here which I will skip. I think I have talked about it in various ways. Let me just show you an example that we have here. So this is again for Mycobacterium tuberculosis because that's what I'm most familiar with, but there are many other cases where Boolean modeling is carried out. And one of the early studies was for understanding Bordetella infection by a group in Penn State, I think Rekha Albert and so on, who also developed this tool called Boolean Net. There are many tools that are available today. In fact, Boolean modeling is also used quite extensively in astronomy, which is what I see, very large systems are modeled by that, and also by the stock exchange thing, the finance kind of modeling. So here we have a host pathogen interactome of Mycobacterium tuberculosis, which accounts for several mechanisms of invasion by the pathogen, defense of the host, as well as the various defense mechanisms of the pathogen. These are what captured in the model. It addresses the following questions. What are the critical bacterial factors responsible for infection? Can we get an idea about that from the data that is known? You can't really claim that the model is complete by any standards. It's extensive but not complete, because it will never be complete honestly. Okay. What are the principal components of the human immune response, and how robust is this response? That's a question you can ask. And how does the outcome of the disease depend upon various parameters such as bacterial growth rate. Now, when does a bacterial growth rate change? A growth rate changes when an individual is treated with a drug. Certain amount of the patient is being treated for the disease. So, the growth rate is much higher when there is no treatment. Growth rate is very low when the treatment is successful, and obviously you have in between values. So, you can have a range of growth rates basically. And how does the outcome of the disease depend on the delay in the onset of adaptive immunity? Again, this kind of models is an individual vaccinated with say the BCG vaccine that is available for tuberculosis, and therefore the adaptive immune system is triggered on much quicker than in a non-vaccinated individual, so on and so forth. You can do that. And are there other cellular processes? Is there a co-infection with HIV, for example? Is there a comorbidity? There is a strong link between diabetes and tuberculosis, for example. So, is there a comorbidity that is coming about, and does that affect? Of course, not all are in the model, only some of them are, but you can ask questions of that kind. How does the system respond to perturbations? When something is perturbed, what does perturbation mean to us? Either there is a mutation in somewhere, some place, and therefore there is a change in the sequence itself, and therefore there is a change in the interaction pattern of that particular host protein to the bacterial, or bacterial mutation to the host, or vice versa. Or there is a change in the innate immune response. The TLR receptor has a polymorphism, and therefore it is very active, or not so active, and so on. That could be a perturbation. Or it could be a chemical perturbation, that an individual is being treated with a certain drug, or could be a vaccine for that matter, and therefore this particular component is up or down. It's modified in some sense. So, that could be a perturbation. One can model all of those things with this model. So, in other words, how do variations in system components such as knockouts or inhibitions or delay in onset of a given event influence the disease outcome? So, the way it was constructed was again do extensive reading of literature to see what is known. So, identification of components, which are the molecules that are linked in some sense to host pathogen interactions, both from the host side and the pathogen side. So, this particular model, this was actually done by my former student Karthik Raman, who is now a faculty member in IIT Chennai here. So, but later on other people have worked on extending this. So, initially the model had some 75 nodes, which has bacteria as one entity, because not much was known about it at that time in terms of molecules, and 18 virulence factors from bacteria, which are, you take the bacteria as a whole and also as 18 virulence factors, 56 different host components in this model, which span about 11 cellular processes and 19 cell types and cell states, and 26 different other molecules outside, these kind of things. Now, the beauty of this is, again, you do not have to say, not all nodes need to be only molecules. Okay, you can have a molecule, but if you do not know what other molecule it interacts with, but you know that it is interacting with a T cell, your second node can be a T cell. That means you are collapsing this entire thing into one kind of a node. There are large number of molecular events that are happening, but you can collapse them all into one node and say T cell, because information is lacking, not because that there are no events occurring. It could also be because the whatever effect that is happening due to individual molecules results only in one particular variation in the cell as a whole. So, it is also meaningful to do it in that fashion. And there is a third reason for doing this, because data is available in bits like that. Some studies are focused, if you look at biology papers, some only talk about T cell states, nothing else. Experiments that are done very rigorously and so on will tell you the change at that level. But some other studies will do a biochemical test and know that there is a particular protein which is kind of loosely linked to T cell state, and so on and so forth. So, the information is really diverse and disparate. This helps you connect them up. So, this is a network assembly which is this, which I will come back to in a second. So, derivation of Boolean transfer function for each one of the nodes, how that works. Initialization of the node state. So, you must have an initial value of 0 or 1. Is this node active or not active? So, for many of them you do have some information. If you do not have, you do multiple combinations, and since you do not know which one is correct, you do it in a large number of 1000 different times. You simulate this and then look at predominant outcome. So, for where it is not available. You can also go back and iteratively and fit the model and say yes, this molecule has to be present for this to happen and so on. That can be done. So, and then you simulate, and in this case we were interested in looking at, this is somewhat like an attractor, 3 different types of outcomes. Is the bacteria getting cleared, like I told you early on? Is the bacteria getting killed and therefore it does not mean anything, infection is cleared? Or is it leading to active disease? Bacteria is proliferating rapidly over a certain rate that we defined as an in-between kind of a thing. So, this is somewhat semi-quantitative in that sense, active disease. Or is this involved in that stalemate and it is leading to persistence and so on. And the rules that we did was we put a bacterial count here, and if it is less than the threshold V min, that means it is cleared. And if I recall correctly, we put a number 1 there, a single bacterium as this thing. If it is between 1 and 5, I called it persistence, and if it is over that, I called it as active disease basically. And this is just a relative scale. You can change it based on the data that you have available to you. And this is the model. Now, each one is a node, you see, and it is connected by edges, and the edges are of different types. You can see colors, some red, blue, green and black. Each one has a meaning, which means is it activatory, inhibitory, is it simply leading to something, or is it two together are leading to this, that kind of a thing, and is it coming from the bacterial system or is it coming from the host system. Those are the colors basically, and these are the outcomes: bacterial clearance, granuloma, active disease, latency, all of these are outcomes. So, that I think I have described the model already. Examples just for you to understand what a transfer function looks like, you do not need to go through all of this. Antigen presentation, which is a very broad function, right. Antigen presentation here is bacteria is obviously required because the antigen is coming from the bacteria, and you have an antigen processing pathway out here. It can be B cell, it can be T cell, both are there in this case. It can be class 1, class 2, T helper cells or T killer cells, whatever, those are all there. And there is some random event because you do not know when it is going to be triggered, when it is not going to be triggered. So, there is some way of, because there are random events, you do this a large number of times and then look for predominant outcome. You can also bias this random event by saying it is random, but the probability is this much and so on. You can put that rule in as well. Whereas now you look at interferon gamma signaling, here the rule is a little more focused on the molecules. So, here you have an activated T cell, you can also have a marker for an activated T cell in terms of a molecule. So, or Th1 cells or Tc cells, it come from any of these things, which means it is CD1R is a particular receptor of T cells and antigen presentation, both need to be triggered, or it can happen with this activated dendritic cells or activated phagocytic cells and so on. Yes.
No way of saying something has to exceed a certain number and only then will a reaction be triggered.
We put that in, but some people do that. So, this is like a very loose threshold. You do have some number of molecules, you can, if you start with an initial population of molecules, you can put a threshold. You can also do it by, if you do it 1000 times and if you see the outcome 500 or more times, you activate this rule, otherwise do not. That is another type of threshold you can put. Okay. So, you can have rules of that kind. Pro-inflammatory cytokines, which is a result of activation of T cells or some other types of cells, are out here. So, you have individual molecules such as TNF alpha or interleukin 1 or GM CSF and so on and so forth, and that will lead to phagocytosis. So, that kind of a thing you have. So, transfer functions. So, in summary, the model parameters are, this was initially done using Boolean net, but there are again number of tools today to do this. So, 3 possible outcomes as I told you, and because there were some random parameters and we also did not know the order, this was an asynchronous update, but we did not know the order of each event, and therefore, you had to do ABC, you also had to do ACB, where you did not know the particular order. So, you do it a large number of times, like I said, and we find that the dominant outcome in all these number of simulations is actually persistence. It is not actual active clearance or proliferative disease, but it is persistence, which fits the clinical picture beautifully. In the sense, you start with many initial states but you end up with persistence. Only some situations lead to disease, some situations lead to killing. That's the message that you get here. And some parameters that were varied here is this delta AI, which is 0, which means mimics vaccinated individuals. This is the delay in the onset of triggering that rule, no active disease. And this is a count of the bacteria, low B naught, which means more persistence, high B naught is more incidence of active disease of some kind. And we have low alpha, low neta values, which are basically treatment kind of a thing. Is an individual being treated or not treated, and therefore the growth rate is changing or killing rate is changing and so on. And high alpha, you get active disease basically because of no treatment, and clearing rate, this is neta, then bacterial clearance is higher and so on. So, okay, that can be. So, you get map state diagrams of this kind. Each little square here is one time point, each little square is one simulation, but it is an average of an ensemble of say 1200 simulations out here. Each one depicts 0 or 1. For this is a node 0, 1, 1, 1, 1, 1 in this case, whereas in this it is over the time course, but your state is defined by a column. So, here I will have 1, 1, 1 and somewhere 0 and 1, 1, 1, so on and so forth. So, do it a large number of times, and then these are the growth curves. It is the bacterial load that we are monitoring here. This is the bacterial count in 3 different states. That is the outcomes that I am looking at. So, what is the pattern that you are seeing? Here the bacteria infection is increasing and going down over a period of time. Same thing here and the same thing here, but in some cases this is persistence, only here it is getting killed upon a certain perturbation and so on. So, and you can do once you have a model like this, you know the influence of each nodes. So, IFN gamma here for example is 0 throughout. If you artificially give IFN gamma, what actually happens to that? Is it triggering the system? And we can actually go back and then verify with known things that are out there. You also have things like phagocytosis, which is a very broad process, which is like a process of some sub-processes that are out there, it's a master process in some sense. And here this is always on basically, because if you knock out phagocytosis, that means there is no way that the bacteria can be killed in any sense, because phagocytosis is very important for anything to happen, right. So, you can do single knockouts and you can do double knockouts. Once you have a model like this, you say this particular process is inactivated and therefore that Boolean transfer function is not activated, and what happens to the system. So, in a large number of them it can still proceed to the last bit and then ends up in persistence is what I have shown here. The colors represent the density of the frequency with which you actually see. This is persistence and this is clearance, and the darker the color represents that a large number of situations lead to persistence. What you will see in a diagonal here will be a single knockout. What you will see, these are the same things, correlation maps basically, same nodes on both sides. This will be when this is knocked out and that is knocked out, which is a double knockout basically. When you knock out phagocytosis or some assembly out here, maturation or something, it always leads to persistence. But when you knock out these inflammatory molecules out here in combination with say this particular one, another cytokine, then it can lead to clearance. When you knock out certain things, it can lead to clearance. When you knock out certain things, it can lead to active disease. That is what I can actually look at that. We have restricted to a combination of 2, which is a double knockout. We can obviously do triple, quadruple, any number of things if you have enough patience and if the question is meaningful for you to do that. But what this can tell you is which component is very important for a given outcome and how you can manipulate the system to bias the outcome to that particular kind of thing. So, that will be the useful thing. And if your initial network is in much more detail with large number of molecules, this becomes very easy to find drug targets of the host or of the bacteria which is interacting with that. So, you can do that directly with that. So, this in a way gives you a bird's eye view of the host pathogen interplay, and this is amenable to various analysis such as node deletions basically. Insights into critical processes in immune response. The same thing goes for vaccination also. If you vaccinate with a particular type of vaccine, right, what is that vaccine doing basically? Is it stopping this process? Is it an antibody and therefore it is interacting with something and so on? Then you can, it is equivalent to knockout. Yeah.
Just one question. I didn't understand where the stochasticity came in. It just is in the specification of the ones and zeros of all of the components of the network. That's where it is.
It is that. Also the frequency with which you can see this often. So, some rules are defined based on the threshold is defined based on how many times are you seeing this thing coming up in the asynchronous updates. So, is a rule being triggered or not triggered to the next state?
I guess one doubt I have about that is that if you have 2 to the 72 odd possible states of the whole network, you cannot sample more. You can make maximum, you can sample 10 to the 2 or 10 to the 3 of them. To what extent, I mean, do you have to weight that sampling or weight those initial states so that they're more representative of, I mean, because you obviously cannot cover all of them, right?
You can't do that. You know, some of them are obviously leading in the early stages, you know, if it is a directional reasonably. Early stages it can only have 2 states, and one state can bifurcate quite nicely and say one always leads to this outcome or the other outcome in terms of the in between nodes. So, you will in some sense, you know, hierarchically eliminate various possibilities at that stage. Okay. So, there are obviously limitations as well to this, because in this case it was 0 or 1, but like I said, people use probabilistic Boolean models and then look at this you know, in a somewhat semi-quantitative fashion, but then it can give you a large amount of insights into critical processes in immune response or a signal transduction pathway, whatever you are looking at. And it's actually a good framework to integrate quantitative data where it is available and then map and then see which one comes out better. So, another type of modeling is rule based modeling. Again, there are a large number of methods for this. One of the things that we used was called kappa, which is somewhat similar to Bayonet gen earlier on. My earliest previous graduate student Soma Ghosh worked on this quite some time ago actually. So, here this is to look at ion homeostasis in, between Mycobacterium tuberculosis and the host. So, to establish itself within the host, MTB has formulated various means of attacking the host system. One such crucial strategy is exploitation of ion resources, as I mentioned earlier, from the host. So, obtaining and maintaining the required concentration of ion becomes a matter of contest. It is a battle actually between the two, because what the host does is to sequester more ion within it. There are storage proteins called transferins, where a large amount of ion is bound to this molecule and it is stored within that, it is put away. And these mycobacteria put out some other molecules called mycobactins, which have even higher affinity for the ion. So, it pulls it away from this storage thing and takes it for itself, because it is after all a matter of competition between affinities. There is another molecule which can take this away, then that is what happens, and ion is removed from the storage molecules and bound to mycobactin instead. Okay. So, obtaining and maintaining the required concentration of ion becomes a matter of contest, like I said, but it is not just these molecules that are involved. The large number of transcription factors and many other events which leads to the biosynthesis of these mycobactins or the ferritins, for example. So, the large number of players on both sides that are involved. Okay. So, systems modeling of ion homeostasis was required because of this. So, a rule based stochastic model of this host pathogen interactions built by integrating information on ion metabolism from 2 different species onto one platform. Like I said, this kind of modeling tool was already available, which is what we used. In this case, it was called kappa. So, relationship between ion concentration and bacterial virulence, as well as the mechanisms by which they encounter each other, was studied. So, it is somewhat like this: an MTB infected phagosome, because there are processes such as trafficking to endosome. This is happening within the host cell, intracellular ion storage compartment, and there is this ferric transferrin bound to transferrin receptor, and some are sent to other cells and so on. And this receptor mediated endocytosis and recycling pathway, all of these are happening out there. And these symbols just tell you the different components: lipophilic mycobactin from the bacteria, and ferric mycobactin, which is bound to the ion then, and you have ferric transferrin and transferrin receptors and so on. So, the model is again somewhat like this: some are bacterial components, some are host components, and some are interactions between the two. In fact, there are very few molecules which are actual interaction was there between very few molecules. So, this is a contact map and influence map. I will just show you that. So, this is a network, and the hub nodes were further grouped together to form bottlenecks. Which is the major bottleneck in this? The bottleneck is something where If you perturb it, it will have the highest influence on the whole system. So that was defined as a bottleneck. So perturbation analysis of the bottlenecks to identify set of events which help control bacterial infections. What I am actually seeking to do is to give a broad overview, not getting into the results as such, because that is not the goal out here. So these are the components: macrophage, TB, and the extracellular compartment which is there between the two. And you can write down influences of each node to each other node in the system, in the sense just by purely by connectivity, somewhat like analogous to your shortest path kind of a thing. Is there some sort of an influence of one node to another through within a reasonable number of steps, that kind of a thing. And that is what you see here. Although there are only this many components, the number of influences become very large, and it becomes somewhat like a cycle, but these are the 4 hub ones which are between the two, which are very, very strongly influencing the whole system. So you see a large amount of crosstalk, indirect crosstalk this is, between the two systems. Okay. And once you build a system like this, the system is built again not based on prediction but known information, basically putting together a curated system. Then you start perturbing each one. So there are some sort of modeled rates for each one of these. Modeled, again, experimental information, but you mapped and you have kind of looked at what is the probable rate between node A going to node B, and that kind of some fitted data is used out there. And you do a perturbation. Perturbation now in a very simplistic fashion can be increase the concentration of that node, increase the concentration to 10-fold, 100-fold, or make it 1/10th, 100th, and so on. Again, increase the rate of transfer of whatever reaction it is doing, change the reaction rate. One is the node size itself, you change it. The second one is the edge size. You can change to again 10-fold, 100-fold, or 1/10th, 100th, and so on. So those are the various perturbations that are done, and it is done on each rule out there. So effect of perturbing each bottleneck is given here. Rule 6 is a molecule called activated IDER. IDER is now a transcription factor. In fact, I do not have slides on that here, but Soma has done further work and shown through molecular dynamics how the states change and how it binds to a third ion molecule, how it binds DNA, and therefore it regulates transcription factor. I think that is just coming out in PLOS, computational biology, that the molecular dynamics work really, that kind of a thing. So you see various things when each rule is varied, what you get, and these bars will tell you the extent of response that you are looking at upon each perturbation, with perturbations on one side, rules on the other side, and so on. So what you can readily tell is, and this is done in 2 situations, non-infection situation, which means there is no bacteria, and an infection situation. I think the red everywhere is infection situation. You see major differences when there is infection situation in certain kind of things. So what you can readily tell is only some bars are showing a high response, right? So only some parameters, some rules are more responsive than other ones. Some are not really influencing the system as a whole. So those form good bottlenecks in that sense. So an example of a bottleneck is when you decrease it by a factor of 1/10, no production of, that is rule 6, which is for IDER. So IDER is not produced, therefore this drastic decrease in the concentration level of ion MTB, because that is indirectly triggering the ion binding to MTB and so on. And likewise you can see bottleneck 5 here, which is a 10 or a 100-fold increase or a knockout both. So here it is decrease in this Fe MTB parameter and increase in this particular thing, or drastic increase in the level of that and no active. So both ways we can see that there is a significant perturbation that is going on there. So this tells us, of course, it helps us understand the importance of iron for bacterial pathogenesis and correlates well with known experimental data that is there. So the transfer in iron complex, which is important for iron uptake and can be used as a possible drug target, which came out as an outcome of this analysis. And dual perturbation and knockout studies gave further insights of which one should be coupled with. Can you couple both a bacterial node and a host node to get a good mechanism going? So the next type of modeling strategy would be graph theoretical kind of methods, where you do not have actually quantitative values anywhere about the rates or what reaction it does and so on, but you can have which protein is interacting with which protein by predicted by a number of different methods. Okay, again information may be available from say something like yeast 2 hybrid studies where, or co-immunoprecipitations, where 2 molecules form a complex and therefore they are known to interact with each other. There are information or data sets actually available of that kind. But you have information about different types, evolutionary information. 2 different genes are always co-located in an operon, or close together in a particular genome or a chromosome in different organisms. In the sense phyletically, there is phyletic conservation between them. So there is a stronger probability that they will interact with each other. And if they are seen with higher frequency in certain phyletic, in certain phyla and so on, then they are considered to be an interaction between them, some sort of an influence between them. There is another kind of method which is actually put out by David Eisenberg and group quite some time ago, is a method known as Rosetta technique, or a fusion kind of a thing, where if a given polypeptide chain has 2 domains in one species, and these 2 domains are present as individual ORFs or individual genes in some other species, they are believed to be interacting with each other because they are close together. Both are present, but in many species they are on a single polypeptide chain, which means there is a reason why they are on a single polypeptide chain and so on. So these things are considered as a reason for predicting the interaction between 2 proteins. Another kind of a thing is co-expression patterns from a large number of microarray or RNA-seq kind of data sets. They are correlated expression values in a different variety of conditions. That means they are important for each other's function, and that is taken as that. So that is another type of thing that is taken. So you can put all these things as if you have a protein-protein interaction networks. In fact, there are for a number of species there are databases where these networks are put out. There is a large amount of noise. One needs to use it judiciously and pull out and curate and so on. So network is nothing but now nodes and edges. So you have vertices. Graph G has V nodes and edges belonging to this whole thing. So neighbors are 2 nodes N1 and N2 connected by an edge. Neighborhood is the set of all neighbors of node N, and you define various parameters such as connectivity, clustering coefficient, degree, betweenness, centrality and so on. All of these things can go on. So you can look at topology statistics like a road network, like I said, which player is connected to which other player, and is a given, does a given node have only one edge and therefore it can only go through that particular thing, or does it have 10 different nodes connected to it, and at point A it can go through one route, and point B it can go through another route and so on, and can we find out which route it is taking. So that is the question to ask basically. A large number of people are looking at networks these days, and you have different types of networks. One type of network is to look at correlation in expression patterns between different genes. Beg your pardon. Then people look at are there modules, for example, is there a city center which is so dense it works like a module compared to a suburb of a city which is loosely connected. It may be connected through one route or 2 routes, but not too many. So each one comes out as some sort of a cluster of its own, but still connected. Okay, you can have modules. Typically a biochemical pathway would turn out to be a module, and the connection between biochemical pathways are the edges between them. There are influences and so on. People also look at motifs. I will come to this in a moment. Are there patterns of linkages between module to module? Again, we can go back to the example of suburbs and the city center or the business hub. So suburbs typically have maybe longer kind of pathways with few kind of hubs between them, that kind of a thing. So and connected only by 1 or 2 routes, whereas there are many more connections that can happen for a very busy part of the city. So these motifs, what is the pattern of interaction or pattern of paths that you get, can indicate some sort of function. So people are also looking at how to compare 2 networks. If I have a network of the traffic pattern of Chennai versus Bangalore, are there commonalities in them? Obviously, when you look at it in the way of city center and the suburbs, there will be commonalities. But if you look at some other kind of a thing, is there one city center or more, or is there IT hubs coming about in the cities, you may see differences in patterns, So both may be interesting. So network alignment becomes an interesting thing to do. Again, a typical question people ask is, what is the shortest route that I can take to get from the airport to IMSC, for example? Is there one shortest route, or are there two shortest routes that are equally viable? And what other parameters that I can put to say that this is the best path that I can take? How do I get from node S to node T given this topology that is out there? And typically biological systems are known to follow a scale free network. So some nodes have very high kind of connectivity and many nodes have poorer connectivity that is out there. So these are all known. I think Barabasi's work and many others have actually looked at it and do that. Another parameter that is important is called clickishness or clustering coefficient. A click, we call it as a click if every player is connected to every other player, right? If you take your laboratory as a node, everybody is talking to everybody, hopefully. So then it becomes a click, right? So, and you can say how clickish is it, clickishness, what is the clustering coefficient that is out there that defines whether it is a cluster or not, whether it is a component, an individual module or not, and so on. So that's something that can be measured. So you can now make a network of host molecules and pathogen molecules. Host pathogen networks can be made, or you can also make it in a more implicit kind of fashion. You're looking at the pathogen in the context of infection in a cell or in a patient or something. You can look at the host, for example, blood profiles, molecules of the blood that you look at, basically blood cells and so on. So you can look at those when a given individual is infected, which is, I'll come to that case study in a moment. So both ways you can do, and also make them explicit. So there are problems with just the topology, which means you do not know which player is active when. To overcome that, gene expression data or proteome data is typically integrated, and you weight the nodes and edges. You can say a node is small or node is big depending on how many copies of this particular protein is available, inferred from gene expression data, which is fair enough for bacterial systems. Take it with a pinch of salt or a spoon of salt for eukaryotic systems, but it still is reasonable because it is relative in scale. And an edge weighting can be done based on the somewhat like a geometric mean of both the nodes connecting the edge, and also based on how centrally located it is in the whole network. Is it, they call that as betweenness centrality. So you can look at measures of that kind as well. These are examples of motifs. You can have directed networks, and that's where most of the work goes in making a network directed, because you need information from literature and curate it and so on. A good example of a directed network is transcription factor network. Our target is not influencing the transcription factor in most situations, whereas transcription factor is influencing its target expression, so on. So you can have directions, and these are common motifs that you see. You can have feed forward, feedback, this kind of motifs that you come across, and some of them are characteristic of certain types of regulation. So that becomes very useful to see. I've already touched upon this. You can also have them in a hierarchical fashion. So people look at hierarchical networks as yet. A large number of tools, for those of you who are just exploring this, that are available that you can utilize for visualizing, for calculating various network properties and perturbing the system and so on. So we use Cytoscape quite extensively in the lab. So there are many other things that you can use. There are some are open access. This particular thing costs a fortune, so we don't really use it, but we have methods that we can do somewhat similar things using open access tools. Okay. This is an example network of asthma, for example. Here the nodes are, some are cells, some are again you have protein molecules such as cytokines, growth factors and so on. This one is just to show you the connectivity that is out there. Eosinophils are triggered by endothelial adhesion, and that is triggered by cytokines, which comes from mast cells and so on. So there is a big complement of interactions that are going on out there. And finally, inflammatory mediators come out. And depending on what sort of inflammatory mediators are there, it can influence the outcome of asthma, basically, that kind of a thing. This model is built with networks, with Boolean modeling, and many other things that are there, many examples for asthma. Again, I think this is just a resource kind of a table, many for curating networks, also for visualizing and so on. So let me not spend too much time on this. I just want to touch upon two more examples, one from our work, one from literature. This is to look at host responses to tuberculosis using a network approach. Now, the question that we want to ask is, so in an infected patient, if you look at protein-protein interaction of the blood cells, taken from blood cells basically. So, do you see a difference in an infected individual versus a healthy individual? Okay. And do you see a difference between an infected individual upon treatment at various time points? So, the microarray data was available from literature, a group in London, basically Anne O'Gara's group, who did the transcriptome analysis at different time points for many TB patients. So, this data was available, which was used for making condition specific networks. Again, one quick point I want to use is when people typically do transcriptome or RNA-seq data, they are only looking for DEGs. They are looking for differentially expressed genes, which is nothing but how many components are up or down in the regulation values and so on. But that does not tell you anything about whether they are connected to each other and whether this upregulation means something to some other thing. The influences are not taken. So, you get that information when you make networks, condition specific networks. Another kind of problem that you encounter with DEGs is it is a ratio. So, if the intensities, in the sense abundances are 150, it is a two-fold change. If it is 1 and 0.5, it is again a two-fold change. So, there is a big problem with looking at abundances. So, that can be overcome by taking actual signal intensities as you read out from an RNA-seq data. And today the technology is reasonable enough for us to allow us to do that. Previously, it was on a relative scale, we could not afford to do that. But today the methods are sensitive enough for us to take signal intensities as you read out from your heat map or whatever of a microarray data set, and then take them as surrogates for abundances of each protein, copy numbers of proteins. Okay. So, that is just to show analogy to traffic networks. So, again I built a large network from protein-protein interaction network of this kind for human cells, a base network which is made condition specific or even patient specific by putting in patient gene expression data or whole blood profile, because that is what can be measured easily. And this network has something like 2000 different molecules, all are molecules in this case, 2000 different molecules with different edges, again curated edges and so on. That is grouped into some sort of logical modules. You will see actin polymerization as one, phagocytosis as another one, protein processing as another one, and so on. This is just a landscape of the model. And when the question we asked is, now there is a typical traffic pattern in the city. Upon infection, where are the changes that are occurring in a completely unbiased fashion? You do not say that it should happen in this module or that module. Where are the changes that are happening in this city's traffic pattern upon infection? Okay, and the city here is your protein-protein interaction network. So, to do that, we computed all versus all shortest paths, weighted by these differential expression values using actual intensities and so on, and then asked for highest scoring paths, the connected sets of paths. So, if there is a node A and it is connected to 5 others, but the change is mediating, the edge is thickened only to B, but not to C or not to D. So, you take AB as the edge, and the AB edge is thickened, in the sense edge is highest weightage is given to AB to E. So, you take ABE as a path, it goes on as a path like that, and all paths within a certain threshold is what you compute. And since the formalism is shortest paths, that is what people generally use, the lower the path score, the more likely the path is likely to be active. That is what is taken. So, you get a large number of nodes and interactions, I will not go into that, and some events that are happening are phosphorylation, ubiquitination, inhibition, these kind of edges are there. Again, just to show that it is a scale-free network, follows a typical scale-free kind of thing, I will not get into that. So, the network is weighted as I explained, and okay, these were the different conditions that were looked at. Treatment of 0 months, which means the patient is just being treated. TB treatment takes a long time, so therefore, after 2 months what happens, after 12 months what happens, and so on. That is what is taken. In fact, I must tell you, if you just look at the gene expression data and do a clustering exercise just by the signatures out there, it clusters beautifully into healthy cluster, patient in the sense disease cluster, and the treatment one is much closer to healthy than to the disease, which is beautiful, which means that the expression variations can capture the condition, it is characteristic of the condition. Only you do not know which gene is correlating. It will be a combination of different genes, that is combination to a particular thing. So, we call this as a response network of pulmonary tuberculosis in the macrophage based on the data that is available, and each of these circles are actually one of the modules that I showed you. Each one can contain up to 20, 30, 40 different genes in the module, and the size of it obviously depicts the extent of activity that we see as we perceive by these paths that are out there. So, another 5-10 minutes is okay, Gautam? 10 is okay. So, what you see here is an overlay of extent of activities through this network analysis in different conditions. So, PTB-M is this one, healthy control is in red. So, you see a big increase in this particular module in healthy control and in this, but in some it is also lowering. So, some modules are being suppressed, some modules are being increased, module as a whole in terms of activity. That means, how many active paths are found in this module, that is what it is saying. Now, you overlay this with treatment, treatment data out here, you see that there is a big change. PTB 0 is red, 2, 12 and HCB, I think HC is laid out in green here, and these are all the 4 conditions. You can see a gradual decrease from TB, active TB to treatment over this, because this is a JAK-STAT signaling thing that is suppressed when infection is coming down. So, that is a known thing which you are picking up. Cytokine signaling also you will see a similar pattern, but something else actually will go up, because as the healthy things, normal metabolism for example, you will see that going up, which is not in this model, but it is done separately. So, you will see some things going up, some things coming down quite linearly over treatment, okay, which gives a good idea of looking at that. Now, I told you that we are only looking at the top activities, highest differential activities in these 2 conditions. Healthy state, same network, no change in the topology, but only activities are changing. You pull out all these active paths in some sense and just visualize them, nothing else has been done that. It turns out that they are all connected to each other, which means these changes that are happening is a concerted orchestrated network of its own, a sub-network of the whole network, which was surprising, it was beautiful that got that. So, there is some meaning to the change that happens here, that is transmitted there, and so on and so forth and so on. Yes.
Some part of this differential upregulation may just be generic response to inflammatory conditions, no? Not particularly TB specific.
Yeah, sure. So, those are also there. Some are TB specific. In fact, some you will see that is common to many diseases and even to cancer. But nevertheless, that is linked to the changes that are happening due to disease. Something must trigger that change, and this shows that most things are connected to each other. In fact, we have done this for several other situations, and we find that they are connected. Small amount may not be connected, but that is because our network may be incomplete or there are changes that are happening. But also randomize the network many times, and we see that such a connected network does not come. You randomize nodes or edges or their weights, and then see that it doesn't, you do not get back the same thing basically. So, these are details of what are the nodes. I do not think you will be keen on knowing that, but there are some which are very well known and many new predictions that come out of this kind of a network. And you can trace them over treatment. See, this is a given path. The same path is shown here in healthy. See, this STAT1 is low out there, and this is 0 months, 12 months treatment, and 0 months it is pretty high, and at 12 months it is becoming the same thing. You also see, and that means it is going closer to the normal state. You also see A to D, a certain path in healthy. A to D is reaching through a different path in disease state, and that is, there are some path reversals that we see upon treatment. Not all will be reversed, I mean there is some plasticity out there, but many things actually reverse, which are good markers for us to monitor the progress of treatment. In fact, that is the current work that is going on in the lab with more network analysis. Because a large number of studies for validation, let me skip that and this thing. Flux balance analysis that I promised to cover. So, I will just go to the slide which shows this thing. So, these are sequence of reactions A to B to C. These are biochemical reactions, typically done for biochemical reactions. And there are some exchange, because no system is isolated, right? It is taking in ATP, water, protons from outside, and it is putting out things. So, there are exchange fluxes that come in and go out of the system. This is written in the form of, say for example, A plus D gives you E plus B, and B becomes C, which is interchangeable, and C becomes something else, and so on. It is captured in a matrix form like this, where A, B, C, D are metabolites, and these are the individual reactions that are happening out there. So, 1s and 0s simply mean the minus sign means it is on the left hand side of the reaction, and the plus sign means it is on the right hand side of the reaction. So, basically we are looking at the flux, right, d by dt of whichever metabolite is now modeled as a stoichiometric matrix S into the fluxes, the relative fluxes of each of the reactions, which at steady state is 0. So, SV is equal to 0. So, we actually eliminate the d by dt term and just take the SV is equal to 0. You also have an objective function in mind, which is for a bacterial cell, what is the purpose of metabolism? You know, it wants to increase its biomass basically. So, you have a reaction for a biomass which is a combination of different things, and it will optimize its metabolism to maximize the biomass. So, you pose it as a maximization thing using I think a common method that is used is linear programming thing, because this will gives you a lot of inequalities, which flux must be 0, the inequality, the constraints basically come from the stoichiometric matrix itself. It also comes from assumptions of mass balance at steady state. So, that is what we actually get. You pose it as a linear programming problem, becomes very easy to compute, and you get a single solution for fluxes for a given condition. And to make this more condition specific, we have a method by integrating gene expression data alone or proteome data, whatever that may be. A large number of variations, as I said, of flux balance analysis, where people actually look at C13 fluxes, data is available for that, or regulatory fluxes, or what we call as integrating transcriptome data and so on. An example from literature, this is not our work, this is for an organism called Francisella tularensis. I think it's a problem as a bioweapon, and there is a concern about that. So, people are looking at this organism. So, this Anu Ranganathan in Pune actually has, while she was a postdoctoral fellow, I think she has worked on this model and got a flux balance analysis of this model. So, the genome was available and its in silico reconstruction was done. In fact, we have a similar one for tuberculosis, but I thought it would be good to include work from another organism, so I put this in. So, the in silico representation includes gene protein reaction relationships. That is the first step of creating a flux balance model. You need a gene, gene to protein, and protein to a biochemical reaction mapping. GPR associations are important. Okay. And some multimeric, sometimes a single gene is not adequate. You require gene 1 and gene 2 to make a protein. You require 2 proteins to make a biochemical reaction, and a given protein can be involved in multiple biochemical reactions. So, it is a bit complex as a picture. So, that is done. Functional classifications of genes included in the model, which just shows you carbohydrate metabolism, nucleotide, and so on and so forth. And this is the overview of what is available. And they actually grew the organism in different nutrient sources to see which pathway is enabled, which pathway is disabled basically. So, these were all given as carbon and nitrogen sources, and you see whether growth is there over a period of time or not there. In fact, there is a high throughput method to do this called phenotypic microarrays, which we used recently for Mycobacterium smegmatis. So, each one is a point in the growth curve basically to see what is the maximum amount of growth that is possible or not possible through that kind of a resource. And now, when they subjected it to flux balance analysis, these are growth curves again with fructose as a source, arabinose as a source, xylose as a source, and so on and so forth, and various validations that they look at. And they notice that amino acid metabolism of a particular kind, tryptophan metabolism, lysine metabolism, all this becomes very important for the bacteria to survive in vivo. These are utilized even when there is lack of a certain carbon source out there. It can utilize, amino acids are usually nitrogen sources, but they can actually utilize this, is one of the outcomes of this kind of a study. So, the relative flux of a given pathway is much higher than what you see in other situations, which means it is a good drug target to be looking at, that you get a large amount of things like this. Once you compute a flux balance analysis of these are called genome scale metabolic models, it actually becomes like a game. You can play with it. You can knock out each node or each reaction, and then you can also change its eating rate, right, in the sense uptake rate of a given nutrient and so on. And you can model a large number of perturbations. We have also modeled effect of drugs, which means one particular enzyme is inactivated by 50%, 90%, whatever. You can put all of those things in and then find out which enzyme is essential, which reaction is essential for the biomass to happen or to occur, and so on and so forth. So, these are some kind of things, and they find that GABA and glutamate influence growth rate distinctly, and those could be targeted perhaps. So, in summary, let me skip this as well. I think there are more examples of viruses. I think I have run out of time. So, let me just quickly summarize. Just one quick thing of showing that people are also looking at structural interaction networks, a smaller number of players, but now the information is coming from structural changes of each molecule to see whether sequence to function and structure to function models, whether that is happening, and whether there are patterns within the cell or between cells and so on. Let me just skip all of this and just summarize by saying that there are a number of different methods that are available for modeling depending on the question and depending on the data that you have and the resolution that you choose to have. But all of these are available, and they give you, of course, insights which are very rich, which can be tested and so on. I think I'll stop here and take any questions that you have. Thank you.
We can take some quick questions and then, yeah, go ahead.
What is a protein metabolite interaction, like in host pathogen interaction? Does the protein come from the pathogen and the metabolite from the host?
You can have all ways, in many different ways. You obviously have metabolism that's occurring in the host cell, also within the pathogen. So, you tag metabolite from host or pathogen basically. You can make bipartite networks, or like in flux balance analysis, you can have two distinct models. Then you can combine them and put what is the interaction between them. Is there an ion transfer function from one to another?
Okay,
so, those will be the connections between them.
Oh, okay.
More questions?
In the flux balance analysis, you showed two kind of different reactions, right? VIs and BIs.
Yeah.
What were the differences actually?
So, B's, I don't remember the names, but B's I think were the exchange fluxes. V's are the fluxes of the actual reaction that are happening. Exchange fluxes means it's either being put out to the system or taken in from the system. It doesn't have to follow mass balance. Okay. All others follow mass balance.
Ma'am, what is the advantage of Petri nets that you showed, like over normal having nodes and connect links between the nodes?
Yeah, those are like more formalized, that the directions are better defined, and there's tokens. So, a given node can change state, which in a network of this kind you either have to make it as a different node, or you can't have two states for the same protein, for example. That's a limitation in this case. Whereas in a Petri net, that is somewhat it's a dynamic kind of a process that you can get.
There are no more questions. Let's just break for coffee. It's outside, and thanks, Suma, again.
Thanks.